Hello everyone, it's a pleasure to be with you today to discuss the next evolution in HPTLC. Today, we are diving into the new visionCATS version 4.2 with its new AI package tools. In this webinar, I'll be focusing on the peaks tool and the classification tool. We will gain some insights into how to configure the analysis parameters and how to use the new tools by demonstrating a couple of real examples. Let's start with knowing the purpose of these tools. As we know, HPTLC has always provided us with powerful visual and chemical data. However, the volume and complexity of that data can sometimes be a challenge to interpret manually. Our goal with these new tools is to bridge that gap, using the chemical profiles to make sense of the HPTLC data without making it hard for humans to guess or struggle with the complex data interpretation. What we will see today is just the beginning. These features are part of a broader strategic roadmap. They represent the first wave in our planned series of AI tools that will continue to roll out in the future. All are designed to make the lab workflow smarter, faster, more efficient, and more reproducible. So, what are these tools about? The current package introduces two core tools, peaks analysis and classification. The peaks analysis is designed to automatically extract key peak data such as peak area and peak height from selected compounds, and then establishes a reliable acceptance range based on high-quality reference data. This provides a clear, realistic acceptance range for chromatographic peaks of a specific compound or multiple compounds. The second tool is the classification, which offers some kind of pattern recognition capabilities. This tool supports two types of learning, supervised and unsupervised. The supervised is mainly used for authentication and assignment. It involves training the model on labeled high-quality samples to classify unknowns with high confidence. Example of uses in detecting adulteration or confirming botanical identity. The unsupervised, on the other hand, is for exploratory clustering, where grouping the samples is based on similarities in their chemical fingerprints. Actually, this is ideal when labels are unknown or unreliable. Before demonstrating how the new tools work, let us briefly recall the three main file types in visionCATS and their rules. visionCATS enables the creation of three types of files: the method file, the analysis file, and the comparison file. The method file is mainly used for the method creation. The analysis file is used to execute the HPTLC runs and acquire the raw data. And the comparison file is used for compiling and evaluating multiple datasets. And here are located the new tools. Chemometric analysis typically require consolidated standardized data, which is why the comparison file in our case serves as a central hub for these tools. I will focus on the analysis and the comparison file, since the new tools require some kind of work before starting to use them. Using the analysis file, we can generate the HPTLC data. The HPTLC analysis usually starts with the samples to be analyzed. And then, using the analysis file, we can execute the HPTLC runs. We need to keep in mind that reliable results begin with consistent standardized data generation. Therefore, all samples and replicates must undergo identical treatment across every step of the workflow, from sample application to detection, in order to minimize variability across the plates. The integrated system suitability test function in the analysis file can help us to verify the method compliance and the parameter robustness. This is very important to reduce any possible misalignment or shifts in the RF values. Once the analysis is done, data can be generated primarily in two ways: high-resolution images recorded under multiple illuminations, and densitometric scanning by the TLC scanner at selected wavelengths. The new tools, whether the peaks analysis or the classification, operate only on the peak profiles. These profiles can be derived directly from scanning, or generated from images by converting the RGB values into luminance-based curves. The generate profile in visionCATS can be used to execute this step. This flexibility allows the users to use existing image data without requiring additional analysis. After generating the profiles, we have to export them to the comparison file. The individual analysis files typically contain data from a single plate. To enable multi-plate evaluation, profiles must be compiled into a one comparison file. To do that, in the analysis file, navigate to the Data tab and use the Export to Comparison function. Repeat this step for each relevant analysis file. That way, the resulting comparison file aggregates the profiles under uniform conditions, ensuring that the algorithms apply consistent parameters across the entire dataset. In this slide, we will know how to access the new tools. Once the individual profiles are exported to a single comparison file, this comparison file will be opened directly. In that comparison file, when we select the profile toolbar, we will see the combined analysis files. In the plot area, we can also visualize the profiles in a 3D projection. The new tools of peaks, analysis, and classification appear directly under the Profile tab. Let us move from the features to the practical application. Here are two examples showing when and how the tools can solve everyday challenges in quality control and authenticity testing. In the first example, consider that you have a food or a cosmeceutical company that incorporates high-quality olive oil as a premium ingredient in its products. And the quality control manager or the lab manager mentions that olive oil's identity, quality, and health benefits rely on its unique chemical profile, especially some key chemical markers which are visible as peaks in the HPTLC profiles. However, due to the natural variability that exists because of different olive varieties, growing regions, harvest years, and the mild processing differences, the concentration of these markers may fluctuate. The key question or the challenge is, how can we define clear data-driven acceptance range for these critical marker peaks to approve good batches quickly and flag deviation? The solution in that case is to use a peaks analysis tool in visionCATS that extracts the peak data such as peak area and peak height. And this will result in defining the acceptance range from the chromatographic peak data of verified high-quality samples. And this will support consistent patch-to-patch evaluation. Here we have 20 fingerprints from authenticated high-quality olive oils. Let's consider that our key quality marker is that one at approximately RF 0.35. After generating the profiles from these images, we can immediately see some variability in peak area and peak height across the samples. So the system will directly define the acceptance range for these peaks, which can be used to accept new batches or flag any deviation. To define the acceptance range of that key chemical marker, we can use the peaks tool. To access this tool, in the comparison file, we need to navigate to the peaks option under Profile tab. Peaks are visualized by default as a scatter plot, where each point represents a peak area or peak height of each sample within the RF range. We can switch between area and height mode from the mode button. Another visualization as a box plot can be enabled from the view option. The desired profile of a specific illumination or wavelength can be selected from the illumination wavelengths part at the bottom left corner. On the right side, there is RF ranges table that provides the detected peak ranges. It assigns color to each range, defines start and end RF, and displays the calculated acceptance value. When a peak range is selected in this table, the corresponding acceptance limits are displayed in the plot as dashed horizontal lines. At the same time, the selected range is highlighted vertically using its assigned color. We can integrate the calculated acceptance range within the limit test function inside the visionCATS analysis file. The acceptance limit, defined with the peaks tool in the previous slide, can be entered manually by the user into the limit test function in the visionCATS, and simply selecting in-range option. visionCATS then automatically evaluates the new samples against this limit. And the system will determine whether the samples are having this marker within the acceptance range and pass the limit test, or flagged as deviated from the acceptance range and fail the limit test. The peaks analysis concept applies far beyond the olive oil examples we have just explained. We can use it to monitor impurities or secondary markers in complex metrics. And so there are many applications like ensuring patch-to-patch consistency in complex metrics like botanicals. Spotting any deviation instantly can be used for tracking product degradation or metabolic changes due to stress or external influences, as so many other applications. Let's have a look at our second example. In this example, let us consider a food supplement company that sells premium American ginseng products like capsules, tea, and extracts. This is a high-value market and a very important medicinal plant. However, the global supply chain is full of adulteration like cheaper ginseng species, lower quality roots, and synthetic additives that get mixed in. Here comes a critical question from the quality control manager or the raw material analyst. Can we reliably detect not normal batches like adulterated or substandard, using only data from our high-quality reference samples? In fact, this is the best scenario for the application of one-class classification, one of the new tools that we have integrated into visionCATS, which learns from the normal pattern from the authentic examples only. And this will result that the system flags anything outside that normal boundary as anomaly, indicating a potential adulteration. Let's go to the classification tool to see how it can help us in this example. We can access the classification tool the same way as peaks analysis tool. From the comparison file, under the profile tab, we can find the classification icon right there. Unlike the straightforward peaks tool, the classification tool requires some setup steps, as we see under the classification steps box, which include selecting the modes, pre-processing, and choosing the models, but we have made the workflow intuitive and guided for the users. The classification steps follow a logical left to right sequence, like the familiar evaluation workflow we have in the analysis files. There are 5 clear steps that lead to the results. In this slide, we will go briefly over these steps. The first step is the mode. The user has to first select between supervised and unsupervised learning. He or she needs to answer the important question: do I have the labels of the samples or not? Or simply, I have the key labels, but I want the chemical data to tell us its own story before applying our own assumptions. The second step is a pre-processing step. It allows you to refine, structure, and select the most relevant data before model training. This involves wavelength illumination selection, data mode, profile tools, and binning. The wavelength illumination selection. This option lets you specify which wavelengths or illumination from a given measurement step should be used for classification. You need to pick the wavelengths where your real samples look different from the fake ones. Multiple selections are also supported. This is particularly useful when different wavelengths carry complementary information. The data mode, it determines how the selected data should be interpreted and used during the classification. There are 2 available data modes: profiles and peaks. In the profiles, we use the full profiles of each sample from the selected wavelengths or illumination. In the peaks, we use only the detected peaks within each profile and their corresponding values. The profile tools, which are mainly used to define the RF bounds, smoothing, baseline, and normalization options. And finally, the binning, and this is a very common term in the multivariate analysis. It groups adjacent data points into a predefined intervals, commonly known as bins. This process can reduce the noise and improve the performance of the classification algorithms. The third step is the dimensionality reduction step. The HPTLC data is high-dimensional data that comprises hundreds of features or variables, particularly when several illuminations are selected from the previous step. The role of this function is to reduce high-dimensional data while preserving the key variants. In visionCATS version 4.2, this tool is only available for the unsupervised mode. Anyways, this step is optional but highly recommended. Available reduction modes include PCA, or Principal Component Analysis, which is a linear dimensionality reduction technique, and it's one of the most commonly used methods. Other 4 additional methods like kernel PCA, sparse PCA, UMAP, and TSNE, which are used for nonlinear reduction. The first step is a modeling or model selection, where you select the model that fits the purpose of your analysis. It's where the actual classification takes place. This step is related to the first step, as under each mode, some specific models or algorithms can be selected. If unsupervised mode is selected, k-means and dB scans will be available. And if supervised mode is selected, one class and multi-class classification models will be available. The final step is the results step, which presents the output of the classification process. The results are typically like score tables, cluster assignments, or classification predictions. We have mentioned in the previous slide that if the supervised mode is selected in the mode step, one class and the multi-class classification models will be available. In this slide, we will know the difference between both classification models. The main differences are, in one class classification we target a single class, while multi-class classification we target 2 or more distinct classes. The one class classification is about answering the question, is this sample A or something else? While the multi-class classification is about answering the question, is this sample A, B, or C? In one class classification, the model is trained on the data from a single class, while multi-class classification, the model is trained on every class. In one class classification, the model learns the normal boundary of the data, and anything that falls outside this boundary is labeled as anomaly or adulteration. But in multi-class classification, it learns decision boundaries that separate classes from each other in the feature space. The main application of one class classification is to authenticate. Example, adulteration detection, outlier detection, substitution testing, and quality consistency. However, the main application of multi-class classification is to assign and predict. Examples like origin mapping, species identification, and cultivar grading. Let's now go back to our use case of the company that sells the premium American ginseng products. It's now clear to us that the best model to use in that case is a one-class classification, which relies on learning the dataset of high-quality samples and can be used to detect the adulteration or outliers. So the logical steps in visionCATS are to first select the supervised mode, and since we work with a supervised mode, we need to assign the good high quality samples to a certain class. In other words, we have to create a class or a group that includes these samples. We can execute this by clicking on the Edit Class option. A new window will pop up. This window enables us to create as many classes as we want. However, in our example, we need to create only one class that includes the authentic samples of the American ginseng. By default, all samples are initially assigned to the unclassified class. So we will create this class and drag the authentic samples to this class, and then we need to save. This becomes a normal training set, and the model later on will learn from it. Proceeding further to the second step, which is a pre-processing step, where we can select the wavelengths or illumination. In this example, I have selected the profiles from the images collected in the white light and long wavelength UV light. I have also set the parameters for the data mode as profiles, and the profile tools for bounds, smoothing, baseline, and normalization. And finally, set the bin size to 0.02. This is a critical step as each parameter or selection will impact the results later. Moving to the modeling step, where we can see the actual classification. Here on the right side, there are the options for the model selection and setting the hyperparameters. We need to select the supervised one class classification and set the reference class as the American ginseng class that we have created in the first step. And finally, we need to select which model to use for the classification. Under the supervised one class classification option, the system offers two different models to choose from. One class supports vector machines and self-organizing map for quality control. For this example, I have selected the support vector machines option. And then we will start training the model, which eventually will score the unknown samples. The plot section will start to display the classification output, giving each class different colors and shapes. Here we can see that the authentic samples are assigned a green color, while the outliers are given a red color. We can notice that some of the unknowns or test samples are given a green color, which means these samples can be considered accepted. In the bottom section, there are some diagnostic plots that give insights into the performance of the model. Example, the jackknife quality control index value, which displays the results of the jackknife cross-validation, and the Hodges-Lehmann null distribution. This chart presents the null distribution of the Hodges-Lehmann statistic. The red shaded region highlights the threshold beyond which the samples are considered outliers. Finally, we land on the last step, the results. In the score table, we will see that each sample is assigned a score. This score value indicates whether the sample falls inside or outside the learned normal region. Green means that the score is on the correct side of the acceptance threshold, and the sample is considered as normal or accepted. While red means that the score is on the other side of the acceptance threshold, and the sample is considered as a potential outlier. These results can be exported as a CSV file. In our example here, 4 unknowns scored green, meaning that this sample can be considered as good samples. To verify the results, I have cross-validated these findings with the raw chromatograms, selected 2 from the red flagged samples and 1 from the green flagged samples. As we can see here in this slide, the green flagged sample displays a fingerprint that closely matches the authentic reference profile. In contrast, the two red flagged outliers show clear visual differences like extra or missing bands, and therefore the image analysis confirms the results that we have obtained with the classification tool. Once the hyperparameters are properly configured and the selected model has been trained, the model can be locked to prevent any accidental modification or deletion. To lock the model, click the Lock Model button. After locking, the button label changes to Unlock Model, indicating that the model is protected. The date and the time of the lock action are also displayed next to the button for traceability. If we later export any new samples this comparison file, the model will directly treat them as unclassified samples, which are then directly evaluated by the locked model. The model can't be retrained or altered while logged, and any attempt to change these settings will give you this warning message. If you need to retrain the model with new reference samples or modify any settings, the model must first be unlocked using the Unlock Model. And that was the second use case example. What I have presented today are just two examples. More example files that use different models and algorithms are available on the CAMAG website for download. Before concluding this webinar, I would like to say that the classification tool offers powerful flexibility with multiple algorithms, wavelengths, and hyperparameters, which is great for advanced users. However, the results can indeed be very sensitive to these settings. Therefore, we try to keep the commonly used values and selection as the defaults, unless the user has more experience to alter them. This keeps the things simple and reliable for most users, while also allowing experienced analysts to fine-tune as needed. Some of the parameters and hyperparameters are also still hidden but are running in the background with default values. However, if any of the users require information about these values, we can provide it to them. We have also created two user guides, one for the peaks tool and one for the classification tool. In these guides, we have explained more about the different parameters and algorithms, which may answer many of the questions about using these tools. To conclude this webinar, I would like to leave you with this take-home message. The new tools are designed to empower smarter HPTLC decision. The main application could be for detection of adulteration and contamination, confirming batch-to-batch consistency, flag degraded, misidentified, or outlier samples, authenticate botanical origin, discover natural groupings or hidden pattern. The target users and customers for these tools: botanical and herbal supplement manufacturers, food and flavor ingredient producer and traders, cosmetics and the natural product care industry, of course academia and research institutes, especially those who are working with chemotaxonomy and phytochemistry, natural product chemistry, pharmaceutical companies, testing labs and CROs, pharmacopoeias, regulatory and control labs. At the end, I would like to thank you so much for your time. I know it might be a dense topic, but hopefully I have made the spectral lines clear for you. I'm now happy to receive any questions, or you can simply send me an email at this email address.